So, good afternoon, good morning. I doubt whether we have any good evening to the members who are joining our. So, good afternoon, good morning. I doubt whether we have any good evening to the members who are joining our. So, good afternoon. So sorry for the glitch. Good afternoon, good morning, good evening to everybody who is joining us today for the second webinar in a series that we started about 2 weeks ago. We are very excited that you're able to join us. We know we have over 500 people who are registered, and maybe others will join as we move. I'm here coordinating from the Joint Clinical Research Center. And again, the history of these webinars, this is incorporated into a project called Capacity, which is funded by the European Union. It's coordinated by Coimbra University in Portugal, but it is a consortium that includes the Joint Clinical Research Center, the Uganda National Council of Science and Technology, the National Drug Authority in Uganda, and Muhimbili National Referral Hospital in Tanzania. So we welcome you all, and we especially welcome our presenter today, Jennifer Adair. She's woken up very early. She's based in Boston. You're most welcome, Jen. A brief introduction about Jen. Jen is a professor and a vice chair in the Department of Genetic and Cellular Medicine. And she's an associate director of the Core Gene Therapy Center at the University of Massachusetts Chan Medical School in the US. That is in Massachusetts, Boston. That's where she's based. Jen completed her Bachelor of Science degree in chemistry at Youngstown State University and her PhD degree in genetics and cell biology at Washington State University. After a postdoctoral fellowship at the National Institute for Environmental Health Sciences, Jen studied gene therapy at Fred Hutch, becoming faculty in 2014. In 2020, Jen was named the Fleischer Family Endowed Chair in Gene Therapy Translation. In 2025, she joined the faculty at UMass Chan to co-lead the first academic department focused on gene therapy translation. Her laboratory develops tools and methods for safe and effective delivery of gene therapy. And very especially, she does think of low and middle income countries. Jen co-founded the Global Gene Therapy Initiative with Dr. Sise Kichio, that is me, in 2020. We are very glad to have Professor Jen to present to us her lecture today. And again, she very much wants to translate gene therapy to low and middle income countries. Jen, you're most welcome.
Thanks so much, Sissy, for that very kind introduction. I am going to try to share the slides here, just making sure you can see those okay?
Yes.
Fantastic.
And what I forgot to say, Jen is a fellow of the Uganda National Academy of Sciences. for 3 years now. Yeah. So she's also Ugandan, but
By marriage, right, Sissy? By marriage, I'm Ugandan. Wonderful to be here with all of you this morning. Today's lecture's focus is on our genes and our health, the very basics of human genetics. For some of you who may be more experienced in the genetic space, this lecture may start a little bit slow, but we'll get to more complex concepts closer to the end. No matter who you are watching this lecture and no matter what your background in genetics is, I hope that by the end of today, you will understand the fundamental concepts of genetics and inheritance, be able to recognize how genetic variation can influence human health and disease, be able to explore interactions between genes, behaviors, and the environment, learn about some cutting edge genetic technologies and their applications, be able to evaluate ethical considerations in genetic medicine, and apply genetic knowledge to your own personal or public health decisions, as well as research and policy. And a key concept, I'll see these little bubbles throughout the talk with different colored text. In this case, the key concept I want to introduce first is that genetics is not destiny. It's simply information that helps us understand risk to make informed decisions. So let's jump right in. Our genetics are based on DNA. DNA is an acronym. It stands for deoxyribonucleic acid. There won't be a quiz. You don't need to remember that long word. DNA is just fine. What's important to understand is that DNA is the instruction manual for all life. If it's the instruction manual, the alphabet that manual is written in is entirely made up of just 4 letters: A, T, G, and C. These stand for adenine, thymine, guanine, and cytosine. Again, you don't necessarily need to remember those names. These are the nucleic acids that make up the DNA. So these 4 letters spell out the story. They are organized in pairs. A always pairs with T, G always pairs with C, and these 2 form a chemical bond, which acts like rungs in a ladder. And that ladder starts to spiral as these bonds are made, forming what we know the DNA structure to be as a double helix, something that looks like a spiraling staircase or a spiraling ladder. If the alphabet is made up of 4 letters, the words are genes. Genes are sections of DNA or segments of DNA which encode for a specific protein. And genes in a genome, the entirety of DNA for an individual, code for all of the functions needed for all of the cells and all of the tissues. And this is quite a bit of information, an entire blueprint for a living thing. And so this DNA can be packaged into the book, or what we call chromosomes. These are structures that organize the genetic library. They serve as storage centers, and they allow us to organize our DNA so that information, genetic information, can be passed on from generation to generation. Now, where is that DNA? Well, it's kept inside of our cells. And the cell is the smallest unit of life, meaning that the smallest living thing on this planet that can be considered alive is a single cell. An example would be a bacteria. Now, all living things have cells and all living things have DNA. Different cells can have different kinds of structures, but all cells have sort of these 3 main features. The first is a cell membrane. This is the outer boundary of the cell. It kind of keeps everything that we call a cell inside. There's a cytoplasm, meaning a fluid-filled space just inside the cell membrane, which keeps all of the components of the cell that it needs to function inside. And then there is another sort of capsule in the middle of the cell called the nucleus. And this has its own membrane. It's called the nuclear membrane. And the nucleus is where all of our DNA is stored. Now, what you're looking at here is a picture of actually human chromosomes, human DNA organized and packaged into chromosomes, as viewed through what's called a transmission electron microscope. And you'll see this image representing human DNA throughout the talk. So our instruction manual is sitting in our nucleus. Well, what makes that instruction get read? There are certain proteins, we're going to call them DNA readers in this case, in the nucleus, and they scan the DNA for those genes, those instructions. They transcribe them into another molecule called RNA. RNA stands for ribonucleic acid, so not deoxyribonucleic acid, but just ribonucleic acid. Now, there are a couple of differences between DNA and RNA. The first is that you might see in that image on the right some of the similar letters, A, C, and G, but now you also see this letter U and you don't see the letter T. One of the chemical differences between RNA and DNA is that RNA is, first of all, one strand, not two, forming a staircase, but just a single strand of letters, and it doesn't contain any T's. Instead, it contains U's, which stand for uracil. Now, this RNA is just like a memo from the instruction manual to the place in the cell where the protein that gene encoded can be produced. So once an RNA message is read, it's actually transported out of the nucleus and into the cytoplasm of the cell. Now, the cytoplasm of the cell is really the factory where proteins can be assembled from these instructions. And this is where you will actually see amino acids, which make up proteins, put together to form specific structures and to serve specific functions in a cell. So this actually entire feature that DNA has genes that are read into RNA, which then get translated into proteins, is what we call the central dogma of biology. Whether you're a living thing that is a human or a plant or a single cell bacteria, this same dogma applies. Your DNA contains the instructions, they get read into RNA, that RNA becomes proteins, and those proteins are what make you function and quote unquote alive. So let's look at an example for a human, and this one I'm going to use for myself. It was just my birthday last weekend, so the example I thought I would use is that I eat my birthday cake. So let's pretend that this cell on the slide is what is known as a pancreatic islet cell. It lives in a section of my pancreas called the pancreatic islet, which is why it's called the pancreatic islet cell. Now, these cells are responsible for making insulin to ensure that your blood sugar doesn't get excessively high. So when I eat my birthday cake, I end up getting a lot of sugar circulating in my blood from that cake. My body will send messages to my pancreatic islet cells saying, hey, there's a bunch of sugar in the bloodstream. The pancreatic islet cell will then read the instructions in its DNA inside the nucleus and convert that to a memo. In this case, if the memo is just a little bit too small for you to read, it says, Jen has once again consumed half a cake on the couch and we desperately need insulin to process the excess sugar. Please assemble and secrete as much insulin as our energy stores can possibly process. So that insulin gene gets converted to a piece of RNA, the memo. Then in the cytoplasm, that memo gets converted into the protein that is known as insulin. And what you're looking at is sort of a three-dimensional cartoon of what the actual insulin protein looks like in humans. This insulin then gets secreted by the cells, and it essentially binds and removes the sugar from my bloodstream so that I am not on a sugar high for an exceptionally long time, or that sugar is not around long enough to do damage to my body. And so that's just one example of how the central dogma can function. And what I've just shown you is the second key insight that I wanted to bring up. Having a gene doesn't mean that a cell will use it. Every cell in a living organism within its nucleus has a complete copy of the instructions, the whole instruction manual for the entire individual, but not every cell uses it. Liver cells use different genes than brain cells, even though they have the same DNA. And I just showed you how a pancreatic islet cell will utilize the insulin gene, but insulin doesn't come from other cells in the body. That is the instruction that is used by pancreatic islet cells. So all living things, as I mentioned, are made up of a cell or cells whose DNA is the blueprint or key to their life, whether it's a tomato, a fly, a human, a fish, a flamingo, a bacteria, a tree. It doesn't matter if it's alive, it has DNA. Its DNA gets read into RNA, gets made into proteins. That is what provides cells their function, and the cells organize into tissues and systems that make up that individual living thing. So what makes us human? You ought to be saying in your head right now, well, it's our blueprint. Our DNA is what makes us human. Now, what's interesting is that we've sequenced hundreds of thousands of human genomes, and what we've found so far is that 99.9% of our DNA is identical between any two humans anywhere on the planet. That is what makes us human. Every human genome, every cell has roughly 3 billion letters of that DNA code in every cell in their body. And this DNA code is organized in humans into 23 pairs of chromosomes, 46 total, encoding about 20,000 genes. And on the bottom right here, you can see what's called the human karyotype. This is just those pairs of chromosomes, pair 1, pair 2, pair 3, pair 4. And what you can see is that we have 22 numbered pairs. And then the last pair on the bottom right are our sex chromosomes. Pair 23. If you are female, you are XX. If you are male, you are XY. And those sex chromosomes are important because we're going to talk about them a little bit later when we talk about how genetics results in traits. Just for perspective, strawberries have 7 chromosomes in octoploid sets. So they have 7 chromosomes and they have 8 copies of each of those 7 chromosomes for 56 total. And they carry more than 100,000 genes in their genome. And dogs have about 39 pairs of chromosomes, 78 total per cell. And they encode about 19,000 genes, which is roughly the same number that humans have. So in the DNA, even though it's only made of 4 letters, can create such diverse information and can be organized in such diverse ways as to create the diversity of life from plants to mammals like dogs to humans. So I want to take a second to talk about how our information results from, or how our complexity as a human species comes from more than just those 20,000 genes. And to do that, I want to illustrate with an example. So here are those 3 humans in photographs that I showed you before, look very different, but we know that their DNA is 99.9% similar. The average height of a human around the world is about 170 centimeters. The average weight is about 65 kilograms, and the average lifespan globally is about 72 years. And I just told you that the average number of genes per human genome is around 20,000 per cell. So let's compare this to a fruit fly. Everybody knows what these are. They're the tiny little gnats or flies that accumulate in your kitchen around fruits and vegetables and sometimes other foods that may be left out too long. Fruit flies are an insect. Their average length is about 3.5 millimeters. Their average weight is about 1 milligram, and their average lifespan is around 45 days. So just mentally, I want you to think about, you see how big and complex humans are. We have 20,000 genes. You see how small and short-lived fruit flies are. How many genes do you think they have? Well, the answer is about 14,000. It's not too different from the number of genes that humans have. So you might say, well, maybe human genes are longer and more sophisticated. Fun fact, about 60 to 75% of the genes in fruit flies are counterparts to human genes. We have similar genes to fruit flies. More than half of our genes are similar to fruit flies. These are why fruit flies are useful in medical research, because they can teach us a lot about how our own genes function. It also means that that complexity can't be coming from just the sophistication of our genes alone. And this is where we can actually look at the total numbers of letters of DNA code in a cell. Humans have 3 billion letters in our instruction manual. Fruit flies only have 180 million. That means that humans have 25 times the amount of DNA per cell compared to a fruit fly. And that means that a lot of our complexity comes from DNA in between genes, the stuff that's not coding for genes. That's what's adding all of the sophistication to our biological structure. I also want to say it is what allows us to be so similar from very different places across the globe. I just said every human genome has 3 billion letters of DNA per cell in their body. And that 1.1% difference in our DNA accounts for all of the variability in humans across the planet, from height eye color, skin color, drug responses, disease risk, and all other traits. 0.1% difference. But let's take a second to put that in perspective. If we have 3 billion letters of code in our body, that means that every person has 4 to 5 million single letter differences compared to any other person on the planet. Now, most of these differences, which we'll call variants, are harmless, but some can influence things like how we taste coriander or cilantro, to how we metabolize caffeine when we drink a cup of coffee or a cup of tea, to how we respond to specific medications that we're given. And different populations can share certain variants, especially if they have lived in a specific region for a period of time. Importantly, understanding that 0.1% variability from human to human and how it impacts human health is the key to a field we call personalized medicine. And we'll talk more about how genetics can be influencing personalized medicine a little bit later on. So how is the genetic information in humans passed between generations? Well, on the left side, we're going to say, here's your hypothetical mother. She got half of her DNA from her mother, your maternal grandmother, and half of her DNA from her father or your paternal grandfather. These are designated by blue and pink chromosomes on the bottom left. On the other side, we have your father. He got half of his DNA from his mother, your paternal grandmother, and half of his DNA from his father, your paternal grandfather. And these are indicated by green and purple chromosomes. Now, in your mother, her reproductive segment comes from eggs that are made in her ovaries. In the ovaries, the first thing that happens is that one of the cells that is a pre-egg, a pre-oocyte, contains one copy of her DNA, actually duplicates the entire instruction book. It makes 2 times the entire instruction manual in that cell. It then separates that into its 2 distinct instruction manuals, but it keeps a mix of your grandmother and your grandfather. That cell will then pull apart and form 2 new cells that have exactly 1 copy of the instruction manual, but it's an equal mix of half and half of your grandmother and your grandfather. Those cells will then separate that instruction manual. Remember, I said we have 23 pairs of chromosomes. What it will do is separate the pairs so that you get essentially just 23 chromosomes instead of 46 on one side and 23 chromosomes instead of 46 on the other. Those cells will then also pull apart, and what you end up with are 4 eggs, each with 23 chromosomes, half of the instruction manual. In your father, the same thing has happened in the testes during sperm formation. Again, the cells are dividing, only instead of eggs, you get 4 sperm. Now, eggs are basically cells. They have a cell membrane, they have a nucleus, they have a cytoplasm. A sperm is really just a very compact compartment that carries DNA. It's carrying those 23 chromosomes from your dad. When your parents got together and that egg was fertilized by a sperm, essentially that egg then became a cell with 46 chromosomes, half from your mom, half from your dad, a quarter from each of your grandparents, and that became you. Essentially, that one cell with exactly 46 chromosomes started dividing and eventually became who you are sitting here today, a mix of both your mother and your father and your grandparents and every generation that came before them. So what we just talked about are inheritance of DNA, and changes in the DNA variants that occur from generation to generation can be inherited, and we call these germline variants. They are present from birth in every single cell in the body because they were part of the original instruction manual. They are passed from parents to children, and you can test for these in blood or saliva because every cell in the body will contain the same variants. But there's another way that we can acquire DNA variants, and that is over our lifetime. We call these acquired DNA variants somatic variants. These develop during your lifetime in specific cells. They are not passed on to your children. And you can't test them just in blood or in saliva. You may actually have to test the specific tissue in which they were likely to have occurred. And you see, I put these 3 circles. There's our inheritance. That's the copy of our DNA code we got from our parents. But acquired DNA changes or acquired variants come from typically environment and behavior. And I'll spend the next few slides kind of discussing some examples of these. So within our environment, we get exposed to certain things. Viruses like HIV actually alter our DNA and can cause changes, somatic changes to the instruction manual. Chemicals like heavy metals, for example, can also induce DNA damage or changes. These can often be found in things like drinking water. In terms of behavior, if you're fair-skinned like me and you spend a lot of time in the sun, you're getting ultraviolet radiation that is touching your skin, and that is causing DNA changes, most certainly in your skin cells. And something like smoking, behavior that induces mutations in lung cells or changes in lung cells are examples of behavior-induced DNA changes. Another key insight is that the age is actually the biggest risk factor for acquired variants in the DNA in humans. The longer we live, the more DNA changes we accumulate. And this is also why certain diseases are more prevalent in older individuals because they've acquired a lot more mutations over the course of their lifetime. Now, DNA variants are genetic differences, which we can't always see or describe unless you have your DNA sequenced. You might have a variation, but you might not know that it actually exists in your DNA. Traits are the measurable or describable characteristics that come from either a single or an accumulation of DNA variants, as well as our behaviors in our environment. Again, remember I told you that 0.1% difference in our DNA accounts for all human variability in height, eye color, skin color, drug response for disease risk. Those things, height, eye color, skin color, those are the traits that we can actually measure or describe. And these can be caused, as I said, by your genetics, by the environment, or a combination of the two. So let's quickly talk about traits caused by genes. There are different kinds of traits. One kind of trait is called a dominant trait. Now remember, you're going to get 2 copies of a chromosome in every cell in your body, one from your mom, one from your dad. A dominant trait means that if you get 1 copy of a gene, even though you'll have 2 copies of the gene, if you have 1 copy for this specific trait, you're always going to have that trait, no matter what the other copy says. And an example of this is having brown eyes. Brown eyes are a dominant trait. 1 gene copy from either parent for brown eyes causes the child to also have brown eyes. Now, the opposite of a dominant trait is a recessive trait. This means that you must inherit both copies of the gene from both parents in order for you to have that particular trait or characteristic. And blue eyes are an example of a recessive trait. You must inherit the blue eye gene from both your mother and your father in order to have blue eyes. And the last kind of trait I'd like to talk about is what's known as a sex-linked trait. Now, here's where we're going to revisit those sex chromosomes, pair number 23, X and Y. Some genes are only encoded on X, and some genes are only encoded on Y. And so a sex-linked trait happens because either the X or the Y chromosome gene is changed or has a variation. And because these genes determine our biological gender, they can influence our traits as well. I've given 2 examples here, males who are XY and females who are XX, but there is a lot of variability in the human genome. There are absolutely individuals born XXX, XXY, or XY that can show or appear to be biologically male or female, but have a wide variability. But one example of a sex-linked trait is red-green color blindness. So if you have normal vision shown on the top, you see a green, a yellow, and a red apple. But if you've inherited variants in two different genes, which are on the X chromosome. These genes are called OPN1LW and OPN1MW. Not important to remember those names. But if you inherit a variant of both of those genes on your X chromosome, then what you see is on the bottom example. Those of you who are red-green colorblind are looking at what you typically see and wouldn't see a difference between these two lines. Now, I said this is on the X chromosome. So if you are male, you only get one X chromosome typically. And if that X chromosome carries the variation, you will be red-green colorblind. But females get two X chromosomes. And so if they inherit one copy of the mutated genes, they will not be red-green colorblind. But if both X chromosomes have those mutated genes, they will be red-green colorblind. And this is why males are more likely to be red-green colorblind than females. Now let's talk about some traits that actually are related to human disease that also fall into these three categories. An example of a disease that results from a dominant genetic trait is called Huntington's disease. This is a degenerative brain disease. It most commonly appears between the ages of 35 and 45. And you only need one copy. If you inherit one copy of the Huntington disease gene mutation from one of your parents, you will have the disease. And so every time a parent who carries the Huntington disease trait or Huntington disease gene has a child, there's about a 50% risk of that child also receiving the Huntington disease mutation. In the middle, we have cystic fibrosis. Now, this is an example of a recessive trait. You must inherit the cystic fibrosis mutation from both your mother and your father to have the disease. In this particular disease, the mutation is in a gene that's responsible for making a protein that breaks down mucus in our airways and other ducts in our body system. So if you have this mutation, you end up getting an accumulation of mucus in your airways and other ducts. This can result in increased bacterial infections, difficulty breathing, and lots of organ damage and other complications. And an example of a sex-linked trait is hemophilia. This is a clotting disorder. Clotting is the process our body uses when we cut ourselves and start bleeding to stop that blood from just continuously flowing out of our body forever, which would not be good. We have platelets and clotting factors that tell our body when we cut ourselves to stop that bleed from happening. One of those important clotting factors is on the X chromosome. And so if you inherit a mutated copy of that, then you are less likely to be able to stop bleeding from happening. And that is why hemophilia is also more prevalent in males than females. Now, skin color is an example of a trait that's actually linked to our environment. Every human on the planet has a melanin gene. How much melanin protein your skin cells make depends on how much sun exposure your ancestors had. Darker skin is more resistant to the damaging effects of ultraviolet radiation because melanin protects that UV rays from being absorbed. So our variation in skin color happens to come from how possibly close to the equator or how much sun we've been exposed to over generations of family before we were born. But it can also be linked to our behavior. Here I'm showing on the bottom a fair-skinned person like myself. If we were to spend too much time in the sun, the red parts of our skin, characteristically called sunburn, are those parts that are massively producing lots of melanin to try and protect those cells from the ultraviolet radiation we just received. Whereas the parts of our skin that were covered have little melanin, and so they appear to be lighter in color. And so this is both, skin color is a trait that is influenced by both our genetics as well as our behavior and simply how much melanin is required from our environment. So now let's jump to an example of a genetic trait that is influenced by the environment in the same way. And the example I'm going to use is sickle cell anemia. Now, the title of the slide says sickle cell anemia versus sickle cell trait, an example of an inherited disease that is influenced by the environment. Sickle cell anemia is a recessive trait. It involves the hemoglobin gene, we'll call it HB. Now, remember, in a recessive trait, you have to inherit a copy from both parents in order to show the trait. Normal adult hemoglobin, we're going to call that A, would mean an individual who has hemoglobin HbAA. They got A from their mom and A from their dad. Sickling hemoglobin, we call that SS hemoglobin, because it's a recessive trait, has to be inherited as HBSS in order for the person to have sickle cell disease. Sickle cell trait, it's confusing because we call it trait, but you can't actually see it unless you measure the hemoglobin, means a person who has one copy of the normal adult hemoglobin A and one copy of the sickle hemoglobin S. They would be HBAS. Now, someone who has sickle cell trait, we can measure it in their hemoglobin, but they don't actually have symptoms of sickle cell disease. The only way they could have symptoms is if they had inherited both copies of the sickle gene, HBSS. I do want to note that there is another hemoglobin variant called C, and individuals can also be HbAc, HbCc, or HbSc, meaning they could have normal adult, C variant, two C variants, or sickle cell variant and a C variant. And this particular hemoglobin variant is most common in West African populations. So if you have two parents who are HbAs, every time they have a child, that's a roll of the dice. Which parts of the instruction manual are going to get passed on, the mother and the father, which half? And there's a chance that the half that contains the S gene will get passed on. And that chance is about 1 in 4 or 25%. So if both parents carry sickle trait, every time they have a child, there's a 1 in 4 chance that that child will be born with sickle cell. HbAc, or the HbC variant, is also a recessive trait. There are some manifestations of being HbCc, but people who are born HbSc also have sickle cell disease. And what is this environmental factor that I talked about that has influenced sickle cell over time? And the answer is malaria. Now, malaria is a parasitic mosquito-borne infection. It does impact red blood cells. When the parasite gets into red blood cells, it causes them to burst. We call this hemolysis, bursting of the red blood cell. In places where malaria is endemic, it's been observed that people with sickle cell trait, HbAs, are about 40% less likely to have severe and complex illness from their malaria infection. This has been described in a number of papers over a number of decades, but I'm just showing one example here, entitled The Protective Effects of the Sickle Cell Gene Against Malaria, Morbidity and Mortality. Now, HbAc can also confer some protection from malaria, but it's not quite as much as HbAs. So imagine that you live in a place with a lot of malaria, and people with HbAs are less likely to be sick, which means they're more likely to be having children during that time. And this means that passing on of the HbA and HbS genes becomes more common in areas where you have high frequency of malaria. So, not surprisingly, in parts of the world where malaria incidence is high, the number of people with sickle cell trait is also high. Now, what's interesting, if we looked at a map of the world and where malaria has been prevalent over years, you would see that the regions of malaria and incidents have changed, and they are continuing to change as global warming is changing where mosquitoes that can carry malaria can survive. In fact, in the southeastern United States, we actually have local transmission of malaria described just within the last 5 years by our Centers for Disease Control. So we can imagine that HbAs may become more common globally as malaria spreads due to global warming. How can behavior change outcomes for sickle cell disease? Well, now that we know about inheritance and how it can be passed on from generation to generation, screening adults before they reproduce gives them as parents the power to understand the risk associated. They would know that each child has a 1 in 4 chance. Knowing that they have that risk can prompt them to seek out early diagnosis, and early diagnosis can support early intervention, and that can definitely improve the outcome of the child's life. Now let's talk about another genetic trait, smoking and lung cancer risk. Now this one, unlike environment and genetics, is influenced by behavior and genetics because smoking is something we choose to do. It's not necessarily something that you're exposed to because of where you live around the globe. But first, before I talk about this, I need to explain a little bit about what cancer is. So we've talked about the cell, the smallest unit of life, and how it's organized, right? And we also talked about how briefly cells are organized in people. Cells make up tissues that make up organs, which form body systems that produce us, the living, functioning version of us. Now, I also told you that, cells have specific parts of the instruction that they use, designated functions, that pancreatic islet cell that makes insulin when Jen eats cake. Every cell knows it has that function, so a healthy cell is doing its function within its organ tissue. When that cell becomes unhealthy and no longer is performing its specific function, the cell will most usually die. This is because in our instruction manual are a whole bunch of safety checks. They are in place to say, are you doing what you're supposed to be doing? Have you accumulated too many mutations? Are you becoming unhealthy? Perhaps it's best if we just kill ourselves as a single cell because then the organism can continue to live and function. These safety checks are also baked into our instruction manual. Cancer is when the cell forgets its healthy function but doesn't die, and instead it reproduces. Here I'm showing that healthy cell on the left becoming unhealthy. It starts dividing, it continues dividing, it accelerates its dividing. At some point, it becomes large. It will start to take over other organ systems like our circulatory system to feed itself a blood supply. It may break off some tumor cells into our blood circulation. Those can travel to other parts of our body and start the division process all over again. That's called metastasis. And this happens because the cell has essentially gotten enough DNA variation to bypass or ignore the safety checks. And it typically does this through errors in the instruction manual or mutations in their DNA. Now, there are lots of different safety checks, and there are lots of different kinds of mutations that can result in bypassing these quality controls, so to speak. So in smoking, we know that the smoke you inhale causes mutations in your lung cells. As you inhale smoke, that damage causes new changes to your DNA. There are, again, lots and lots of studies demonstrating this. I'm showing an example of one study that specifically was done in the United States that related from a large number of patients influences of behavior and age and how long a person smoked with their likelihood of developing lung cancer later. And some general themes from this particular study highlighted that these mutations caused by smoking can directly lead to cancer, and that actually the number of years that you spend smoking is more important than the number of cigarettes you smoke per day. Two people who smoke for 20 years each, one person who smokes exactly one cigarette a day, and one person who smokes a pack of cigarettes a day still have approximately the same chance of developing lung cancer later. Again, those who are older or who have inherited risk factors have an increased risk of lung cancer. If you're older, you've already acquired more mutations in your lungs, and so your chance of getting cancer is higher. When we talk about inherited risk factors, this would be something like perhaps I was born with a germline change, again, one I inherited in one of my safety checks already. And maybe that safety check is in my entire body, or perhaps it's just relevant in my lung cells. I don't know I was born with that mutation, but it is there. And if we were to sequence my lung tissue and know that that safety check was already off, then it's fewer mutations required for the cell to become able to bypass its safety checks and become cancer. And my risk of lung cancer would be higher. But we know that not all of the longtime heavy smokers get lung cancer. And so why is that? Well, when these researchers looked in the lungs of people who smoked for a very long time, they did always find more mutations than a person who was healthy who did not smoke. But in some individuals, their lung cells actually acquired mutations that suppressed the cancer-causing mutations. Sometimes it boosted the safety checks. So the types of DNA damage that you can acquire can sometimes have the opposite effect than what we think. In this case, again, just as the example, they inherited mutations that perhaps increase the possibility of their cells safety checking the integrity of their DNA instruction manual. And this prevented them from getting lung cancer in the long term. So behavior can also change lung cancer risk. If you stop smoking earlier and shorten how much time you smoked, you greatly reduce your risk. If you're going to continue smoking, monitoring your health to make sure that you know how you're feeling and can catch lung cancer as early as possible, but also considered other dietary and lifestyle changes that can improve your outcomes, such as eating a diet that boosts your safety checks in all of your cells. All of this to say that your genetics are like the hand of cards that you've been dealt at birth. Your environment and your behavior is how you play that hand to determine what your overall health outcome will be over the course of your lifetime. And always, family history is the best predictor of your risk for disease because families share genes, behaviors, and environments. And so they can provide a lot of information about how your genetic hand being played may influence your health outcome later on. But it's also important to know that having a genetic risk factor does not guarantee that you will get a disease later. It also means that not having a genetic risk factor doesn't mean you won't get the disease later. Environmental factors, lifestyle changes, and early intervention can significantly change outcomes. But again, genetic risk is not disease inevitability. And this is where genetic testing provides us knowledge, and that knowledge is power. I'll briefly discuss 4 kinds of genetic testing. The first is diagnostic testing. This would be testing to confirm that you have a suspected genetic condition, either because you're displaying symptoms or because your family history suggests that you may be at higher risk. Predictive testing is assessing your risk before symptoms appear. It can also be because of family history, or it could simply be because of an exposure event or because of general concern. Carrier testing identifies carriers of recessive conditions to help inform reproductive decision making. And then the last kind of testing, which we haven't really touched on, is called pharmacogenetic testing. There are, while we all have the same 20,000 genes, give or take, the ability for us to process specific drugs can vary widely depending on how much of our proteins that are responsible for metabolizing those drugs or using those drugs are made by our individual cells. And so pharmacogenetic testing can actually look at how much drug should be given based on how much protein you have relevant to whether or not that protein has a DNA variation in it or not. But important questions that any human should consider before doing these kinds of testings are: what can or will you do with the information? How might the results affect family members? Are there medical management options available? And would you want to know if there are not? And what are the psychological implications of having this information and knowing it? Some people feel very empowered by knowing their genetic results. Other people feel absolutely limited and afraid then of doing anything in the world, behavior or environment wise. And you have to decide where your psychology falls on that spectrum. But there are some moral and ethical challenges of human genetics as well. There are 4 I'll discuss: privacy, discrimination, equity, and consent. Privacy: who owns genetic data? Some of you may have read in the news, there was a company in the US that would screen human genomes called 23andMe for 23 chromosomes and know who you are. They went bankrupt, and there was actually quite a large legal case in the US over whether or not other companies could buy that genetic data. So who owns genetic data? Is it the individual from whom the genome came from, or is it the body that did the sequencing? Discrimination: can any of your genetic data be used against you? Could a future employer not give you a job simply because you carry a genetic risk factor? Or would that be considered discrimination, genetic discrimination? Equity: will advancing genomic data increase or decrease health disparities? And we'll talk a little bit more about that on the next slides. And then consent: how do you handle genetic data that affects families? If you learn, for example, that a parent carries Huntington disease, are you obligated to inform the other parent, or do you leave that to the two parents to decide what to do when you know that any child they might have has a 50% chance of inheriting the Huntington disease mutation? And nobody really knows the answer to these questions yet, but there's something that we need to start conversations around to understand how society wants to handle these issues and come to a consensus on the best ways to move forward for our specific populations. Now, equity in human genetics is a major barrier because not everyone has access to the same level of genetic testing or screening or the same medical interventions for the same disease condition. Using the example of sickle cell, we know that hydroxyurea early on can definitely improve outcome, but it's not necessarily available or adopted by many cultures around the world, even though HbAs carriers are highly prevalent. And all of this genetic information related to human health is specifically pulled from a more defined set of groups. There are some groups that are very underrepresented in the human genetic data that is guiding modern medicine right now. And the example I will give is primarily African populations. As of 2021, of the hundreds of thousands of genomes I told you that have been sequenced around the world, 86% of them have come from Europeans, and only 1.1% have come from Africans. So we do not have adequate representation of the entire human population in our genetic data. If 86% are Europeans and 1.1% are African, that means that the other 13% represents all other genomes in the world across all different populations. Now, interestingly, just that 1.1% of African genomes was enough information for us to see the story of global human migration, with all humans originally coming from the Rift Valley in East Africa more than about 150 to 200,000 years ago, and then migrating to all of the other parts of the globe. Imagine what we could learn about global human health if we increase the diversity of genetic data to include more relevant populations, including the ancestral population of Africans. And there are lots of new technologies that are emerging to help reshape how we think about human genetics and human disease. Functional genomics, polygenic risk factors, liquid biopsies, gene therapies, and artificial intelligence or AI-powered diagnostics. I'll spend about one slide just really briefly telling you about each of these things, what they mean, and how they are shaping how we think about human disease. Just because you have a gene doesn't mean that gene necessarily will cause a trait or a characteristic or a disease. Functional genomics is the field of study that bridges the gap between genetic variants and therapeutic interventions. It actually takes your genomics and how genes are regulated, your transcriptomics, all your RNAs and how they're made, and combines that with what proteins they make and what functions those proteins serve, shown as metabolomics here, and how all of that contributes to actual phenotypes in humans so we can understand which gene variants actually matter. Polygenic risk. Polygenic stands for many genes, many genic genes. Most human diseases don't come from a single gene. They actually come from a large number of genes interacting in complex networks. So polygenic risk actually uses computer algorithms and other components, as well as human health data, to combine thousands of genetic variations across thousands of individuals and predict which ones are actually at the highest risk for acquiring a disease trait based on that inheritance. Liquid biopsies. Now, we talked about cancer, and we said that, cancer can come from acquired mutations. It can also come from inherited mutations. But in acquired mutations, you would have to actually biopsy or test the specific tissue in which the mutations occurred. But there are a number of groups who realize that when tumor cells move around the body, some of them do die and they release their DNA or RNA into circulation. And so they've developed more sensitive technologies that can detect tiny amounts of cancer DNA or RNA circulating in the blood from just a few dead cancer cells to actually determine whether or not it has cancer. A blood draw that can find cancer anywhere in your body would be a pretty powerful tool, since most of us at some point in our life have had some kind of blood test. Now, gene therapies, which are my field of study, are certainly emerging. We have about 33 different gene therapies approved around the globe right now. Most of those are authorized in the US or Europe. These are actual approaches to change the central dogma, to actually reshape the DNA to RNA to protein to combat human disease. And I'm showing a toolbox on the left because the number of tools that are emerging to be able to make specific changes to DNA or RNA are massively increasing over the last 2 decades. And this gene therapy or rewriting the genome is going to become an integral part of how we treat human genetic disease moving forward. And then lastly, artificial intelligence powered diagnostics. This includes things like machine learning to process lots of genetic and health data and clinical imaging data all at the same time to more accurately diagnose diseases in a more rapid time frame. Again, functional genomics will feed into AI-powered diagnostics. And so increasing studies on both parts of the spectrum are important to make sure that we actually do this meaningfully and mindfully. And all this to say that the future of personal medicine is coming. This is the point where your genetic makeup would be considered along with your environment, your lifestyle, and any specifics of your condition or behaviors or disease status to essentially ensure that you get the right drug at the right dose at the right time to maximize outcome for any given disease. And this is where we would like to go, but we really want to make sure that it's not done for just a segment of the population, but actually includes everyone who could benefit from personalized medicine and information from their genetic makeup. And so with that, I will stop. I thank you very much for listening, and I'm happy to take questions if there are any.
Thank you, Jen. We are clapping. We are not online, but we are in the boardroom with 8 other people. Thank you very much for simplifying genes, the basics of genes. And I know there are several questions now already in the chat. And we will read them out so that you answer them. Although we also want them documented, we want to post the questions for people who may not have been able to attend. But as Jane we will have about another 30 minutes for her to answer questions. There is a form that has been shared by the IT. Please fill it out. We want to evaluate what the webinar presentation has been, what you have learned, what can we do better in the future. Should we call back Jen? Also, you can put your comments there. Okay. Do you want to read the questions, Annette?
All right. So the first question, before the Human Genome Project was completed, the best estimates were that humans have 80,000 to 150,000 genes. Once completed, we found that we have approximately 20,000 genes in the genome. What was your reaction when the much lower number of human genes in the genome was confirmed? Did it change your perspective as a genetician at all? If so, why? If not, why not? That's from Adrian.
That's a long question. Yeah.
It's good. I will say, I often tell people I'm a child of the Human Genome Project because I was in about seventh grade. I was 13 years old when they started sequencing the very first human genome. And I was in my second year of my PhD when they finished just the very first one. 2001 is when the first human genome was sequenced. And there was a lot of wild prediction about how many genes we would have. I think everyone was quite surprised to learn at how few we had. The relationship between our genome and that of a fruit fly, that was one of the first fun facts I learned in my PhD, is that, we had almost the same number of genes as a fruit fly. Most of those genes were similar, more than half, but we somehow had massively more quantities of DNA in our genome than a fruit fly does per cell. And that inspired me. I found it absolutely elegant and amazingly beautiful that much complex biology could be packed into that much DNA. And it certainly made me interested in studying how the DNA impacts human health, and eventually led me to gene therapy, which is how we can use that information to actually create meaningful therapeutics for individuals.
So before they read the next question, I have a quick question. You had the introductions for Jane, what she has done. She started with biochemistry. She's now a gene therapy expert. We are trying to build a cadre of people here that can engage and bring DNA to disease, translate DNA, what happens to disease. So maybe you can also give the people online, it may be late for me to go and start doing gene therapy, but what do you advise their young people online, their older ones like me, in terms of training? What do you see as roadmaps from wherever people are?
I would say, webinars such as this are a great place to start. There are tons of online resources. I referenced a bunch of them. You don't necessarily have to read a complex scientific paper. YouTube has a tremendous number of resources to start learning about gene therapy and human genetics. If you just do an internet search for Gene Therapy 101 or the basics of human genetics, you will find other lectures and other materials to support your learning like this. Practice your science and your mathematics in school, if you are able to do so. If you are in undergraduate study or thinking about going into your undergraduate study, biology, chemistry, physics, mathematics, or even computer science are all entry point fields. Medicine is certainly another place to carve a path into this space. Eventually, if you want to do what I do, which is build new technologies, you need to get some experience in a research laboratory. And there are lots of places all over the world, but actually, Dr. Sissy is starting a gene therapy research at the JCRC, along with a number of other institutes. And they also have a training program specific to gene therapy as a possible cure for HIV that is going to be recruiting master students, PhD fellows, and MD fellows that I highly recommend reaching out to Sissy or other members of her team to find out more if that is of interest.
Thank you, Jane. We've already sent out the adverts. I think they went out today. Or
Great.
if they did, they should be also on the HCRC website for people who are serious and want to apply and join this field. Yes.
So how does HBCC manifest in comparison to HBSS?
That's a good question. So I mentioned that the C trait is also a recessive trait. So you have to inherit both copies. Remember that C also gives a little bit of resistance from severe malaria infection. So in places where C happens, people with AC become more and more prevalent because people with HbAc are going to have a slightly better outcome from malaria than people who are HbAA. So that's how you get a large number of HbAc carriers in one space. If you have two parents who are HbAc that get married and have a child, they have a 25% chance of that child also inheriting both C copies and becoming HBCC. C is a different mutation than the sickle mutation. And what that does to the protein is different than what the sickle mutation does. So a hemoglobin protein is going to form a certain structure, and its job is to carry oxygen in the red blood cell. If you have the S mutation, you form that structure, but it also causes the proteins to want to stick to each other and form these long chains called polymers. Polymerization is what we call that. And that long chain is what causes the sickle cell to become very rigid instead of flexible. And so that's what the S mutation does. The C mutation causes just a little bit of change in how the protein is structured, and it makes it just a little bit less good at carrying oxygen. But again, since it's a recessive trait, if you have AC, you don't see it because A is the dominant trait. But if you have CC, then you will have it.
All right.
Then another question is about the diagnostics. You talked about liquid biopsy. You talked about AI-powered diagnosis. Do you have any examples of which conditions can be diagnosed as such? Because we want to bring some of these things from your country to local here, Uganda.
Yeah, so there are a couple of companies in the US. I'm totally blanking on the name of one, but I can certainly look it up here in a minute, that have developed essentially what are called cell-free DNA. Cell-free DNA just means DNA that's not inside of a cell. Instead, it's just free-floating. And they've developed panels of mutations, both acquired and inherited, that are common in different kinds of cancers. So you can say that, first of all, it was tons and tons of patients who were positively diagnosed with, let's say, ovarian cancer or lung cancer or pancreatic cancer. And they gave tissue samples. Those samples were sequenced. They looked at how many mutations occurred, where they occurred, and they found over lots and lots of people mutations that very commonly were associated with that particular kind of cancer across many, many people. They developed assays that allow them to take a blood sample and then assay for those specific mutations to just predict whether or not there would be risk. So remember, these assays are based on a cancer cell somewhere in your body dying and releasing its DNA. That DNA is now free floating. If they find free floating DNA in your blood that carries some of those mutations that they know are associated, it would be a prompt for them to do more diagnosis to look for, say, ovarian cancer or pancreatic cancer or lung cancer. I don't know the full spectrum of cancers that are included in those chips, but that is one example of how liquid biopsies are being used to detect cancer early. In terms of AI-mediated diagnostics, right now, there's a tremendous amount of work looking at radiological images and combining that with other information. I'll give the example of breast cancer. The most affordable and easiest form of screening for breast cancer is a mammogram, but those mammograms have a potentially high frequency of false positives, meaning that they'll detect things in the mammogram that are not essentially cancer, and then that requires additional screening to move forward. So they are looking at AI, essentially interpreted radiologic images, to combine lots of different data on age, health, possibly also genetic data, to say that this person is very likely, yes, to have breast cancer and should go forward for more advanced screening or a biopsy versus not.
Thank you, Jen.
So the next question, in the case of malaria, does it mean environmental factors don't have to directly alter DNA, but could simply be a facilitator of propagation? In this case, do we still call it an environmentally linked gene?
That is a great question, and it's subject to interpretation. Just like the total number of genes in our genome changes, the more sequences we get, and the more individuals we look at, the spectrum of possibilities changes. I would say that yes, the answer to the first part of your question is environmental factors don't have to directly alter the DNA, but can be facilitators or propagators. And we would still call this an environmentally linked gene, but it can also be a hereditarily linked gene. So for example, within certain populations of people in certain religious groups. And one example of this would be people of Jewish faith. They often tend to intermarry with other people of Jewish faith. And within the Jewish population, they used to reside in an area where there was high malaria. And so there is a high potential for someone of Jewish faith to also carry the sickle cell trait. And so if you marry within your same community, then your risk of passing on the sickle cell trait is also higher.
All right. May you please elaborate on the risk of passing on mutated genes to offspring among aged male and female parents?
So remember that the acquired mutations happen in tissues that are typically exposed to behavior of the environment. Eggs and sperm tend to be fairly protected, but it's not, a complete thing. We do know that the risk of genomic alterations in offspring increases with age of the parents. For example, there is a genetic trait called trisomy 21, meaning you get 3 copies of chromosome 21 instead of just 2. And this has to do with when the cells are forming and they're splitting those pairs of chromosomes. Instead of splitting chromosome 21 into 2 different egg or sperm, you end up getting both copies in one. And so then one parent provides 2 copies of chromosome 21 and the other parent provides 1 copy. Trisomy 21 is what results in the trait we know as Down syndrome in humans. And so in people with Down syndrome, they actually have 47 chromosomes because they have 3 copies of chromosome 21. And we know that the risk of trisomy 21 increases in parents who are over the age of 35. And so that's one example of how that older parents can have an increased risk of transmitting different genetic variations to their children.
Right. Thank
But
you.
again, it's just a risk. It doesn't mean certainty. There are lots of people over the age of 35 who have kids who do not have trisomy 21.
But the warning has been sounded.
All right. Sicklers or sickle cell trait individuals tend to attract each other. Do you think there is a genetic predisposition that influences this behavior?
I don't know that there is a specific behavior associated with having sickle trait. I think it's simply just that in certain parts of the world, the population just has such a high proportion of people with AS that the likelihood that you're attracted to another person with AS is high just because most of the people are AS. I haven't yet heard about people with AS and AS being attracted to each other for other biological reasons. But I would also say, there's so much we don't understand about human behavior. Our genetics is so complex. I told you, we just sequenced the first human genome in 2001. We've now got hundreds of thousands of genomes, but we still don't know exactly how many genes. That's why I said approximately 20,000. We're dialing in on that number, but it's still uncertain. We have not yet approached the full spectrum of what is possible. There are lots of people trying to study how genetics functionally relates through proteins and metabolomics and all those other things to how we choose mates to reproduce with. And I am not an expert in that space, but I know that it's complicated. And that there are many things we still have yet to learn.
Right. In regards to privacy, can a parent sue a company for using their genetic data without their consent in case their child participates in a genetic study? This is because they both hold the same, like the child holds all their genetic material.
Yeah. I would say, there are different legal ramifications in different geopolitical borders around the world. The example I gave of that company, 23andMe, filing for bankruptcy, There was actually a legal case in the US that went all the way up to Congress and the Supreme Court called GINA, which was the Genetic Information Act, which says that you cannot indiscriminately sell human genetic data to other resources. There are similar legal frameworks in place in Europe. So one would be understanding what the legal framework in your area would be. But I think a general strategy that I would advocate for is the person who provided the genetic material should always be consulted about it. If that person has already passed on and can't be consulted, that's a different story. But again, asking those questions like, what are you going to do with the information? How will it impact the family? What are the psychological ramifications of this? And then are there medical outcomes? Have to all be considered. So I would be thoughtful about it. And I would just say, I would always essentially consult where possible to understand how everyone feels about it. There's a mutation we screen for here in the US called BRCA12. This is a gene that very specifically impacts families and increases the risk of women in that family to get breast or ovarian cancers. And there is a lot of discussion. You can read news articles about it. The famous actress Angelina Jolie is a carrier of this mutation and lost her mother very early to cancer. Went public in an op-ed in the New York Times about her decision to get tested and then how she decided to move forward and protect her health after that, which you can read online. These are very personal decisions. So when I say the future is personal, I mean, yes, you can use genetic data to find this, but you also get to make a personal decision about what you want to do with it. And being the steward of your own genetic data is part of that conversation.
That's interesting. From the smoking study that you shared, did the researchers sequence genes responsible for lung cancers and compare them with smoking history or metadata? And if so, which genes did they target? Also, how did they handle linkage disequilibrium in terms of races?
It's a very good question. So I'm going to take a second to answer some of the question, but then also explain a few of those terms to folks who may not be as familiar. So did they sequence the genes responsible for lung cancers and compare that with smoking history data? And the answer is yes. So they took lung cancer samples, sequenced them for total variations, and then performed an association study between those mutations and the patients who developed cancer, as well as patients who were long-term smokers who did not develop cancer, and compared those to a pool of healthy people. So a lot of those lung samples were come either from other procedures that allowed them to collect tissue from the lungs or from patients after death who had donated their bodies to contribute to scientific knowledge. And so that's how they know for sure that there were more accumulated mutations in the long-term smokers versus the short-term smokers versus the healthy population. And the metadata, meaning all of the data associated with the population's behavior, their environment, their genetics, and their condition was put together to make that comparison. There have been more studies. That was a 2003 study. It was pretty old. There are more studies since that have better pooled this information together as well, but that was sort of one of the first studies that clearly explained the relationships that I was talking about in that slide. They didn't target specific genes. They actually just did an open sequencing to say any variation needs to be considered as potentially contributing. But they did find common mutations. And they also found common mutations in those people who were long-term heavy smokers that did not develop lung cancer. One example was a gene called XRCC1. Not important, the letters and numbers. That's a gene that is definitely involved in safety checks. That gene is responsible for a protein that will repair damage to DNA. And so what they found in those individuals who acquired the XRCC1 variation is that they accumulated mutations over the time they smoked, and then those mutations just plateaued off because at the top of that hill, they acquired the XRCC1 mutation that made it more active, and it worked twice as hard to then essentially plateau the number of mutations. They did not perform linkage disequilibrium in terms of races that I am aware of in that paper. It may be buried in supplemental data. They were very specifically saying that the majority of people included in that study were Caucasians who smoked in the US. And that was generally because Caucasians were actively seeking medical care more often in the context of the place in the US where this particular study happened. And so these are ways that we have to think about genetic diversity and population variance and how it may contribute.
Right. Please help clarify further on HBC in sickle cell genetics related to HBAS and HBAA as far as disease severity is concerned.
Sure. So I will say that I'm not an expert in this particular space, but what I understand is that so HBAA is adult hemoglobin, carries a certain proportion of oxygen, and it has these nice little round red blood cells that bounce down the vessels. HBCC is a red blood cell that's still going to be round and bounce down the blood vessels, but its oxygen carrying capacity is going to be reduced compared to AA. AC is going to look like AA. SS is going to be a red blood cell that no longer is red and bouncing and round down, or sorry, not from red. It's no longer round and bouncing down the vessels, but it has this like stiff sickle shape that gets caught in the microvasculature. HBAS is also going to look like HBAA. So HBAA, HBAC, HBAS look like HBAA, HBCC, round red blood cells, a little bit lower oxygen carrying capacity, HBSS, sickle-shaped red blood cells that also have the capacity to block blood vessels. And then CS is going to look more like sickle. So you're going to see essentially that reduced oxygen carrying capacity and some deformation of the red blood cells. That's my understanding of how that works. I hope that clarifies. If I'm not getting the question correctly, please feel free to ask again too.
No, I think you have explained it very well, but this brings me to another question on diagnostics. We have now point of care diagnostics. I don't know if they pick the HBC, although I think the full HB electrophoresis would be able to pick that, but it's not also very easily accessible to patients who want to test themselves. Did you hear me, Jenny?
Yes, I can hear you. And I would say I agree. So generally across the world, even in the US, you know, we have rare disease conditions. So things that only impact a certain population in a certain geographic area, they're not as common as the other variants. And so it's really difficult to get screening access for those variants as well. When you're talking about a test that's going to specifically look at, for example, hemoglobin. And so we also have this discussion here around rare and ultra rare diseases and genetic variants associated with those. I think, you know, taking a more, again, objective approach and saying, let's just understand all the variants. And if we're going to provide screening or diagnostics, provide all of the variant screening options is useful. We think right now that the primarily HBC hemoglobin is inherited in West African populations, but if we increase genomic diversity around the world, we may find that it actually does exist in other groups of people, in which case it would be more important to screen more generally instead of specifically.
Okay, there are also some hands. I think we'll allow some people to ask their questions. Although there are more questions in the chat. Who is that?
Joyce.
Joyce, you can go ahead and ask your question. Joyce, you can unmute her. She's not muted.
I think her hand just went down.
Sorry,
Oh, wait,
I
here she
already
is.
asked in the chat. Thank you.
Okay. Thank you, Joyce.
All right, so I think we can proceed with the questions in the chat. How would safety monitoring of gene therapy look like?
A good question. It depends on the gene therapy. There are some gene therapies that actually just modify the DNA to RNA to protein without changing the instruction book in the nucleus. And so for those kinds of gene therapies, you may want to just monitor disease, overall outcome. Did we improve the disease? Did we not improve the disease? You may want to check to see if any of that DNA that is actually in the cytoplasm is gone into the nucleus. But it also depends on the dose that was given. For kinds of gene therapy where we actually rewrite the instruction book in the nucleus, it's really important for us to look at things like how accurately did we correct the right section, the right word in the instructions versus accidentally create changes or typos in other parts of the book. And so that we can sometimes do outside the body with a blood sample, but not always. Importantly, in gene therapy, if the cell you're trying to get the gene therapy into has to divide, like blood cells, for example, have to divide in order to keep supplying your body with blood throughout your lifetime, we have to get it into the instruction book. It has to go into the nucleus, or every time those cells divide, they'll lose it. Whereas for other cells, let's say like cells in your retina, in your eye, maybe we want to correct an inherited mutation for blindness. You're born with your retinal cells. They're not growing, they're not dividing. So we know where exactly to put the gene therapy. We're not going to go back into your eye to monitor that because that would be super invasive. But we can monitor vision and we can look at other parts of your body. But in your blood, we can also take a blood sample to actually monitor the gene therapy and how well it's going. So it's a complex monitoring system that involves whether or not the patient is feeling good and we've seen an improvement in their disease, or whether or not they've developed new symptoms for something else that require us to take a harder look at whether or not the gene therapy may or may not have been responsible. And then there's, potentially monitoring in blood-based gene therapies for just how the blood cells are performing and what that change in the DNA actually looks like over time.
Yeah, quick question, Jen. You talked about somatic changes, genetic changes. Can you briefly talk about DNA and why do we even talk about it in HIV space?
Yeah, so HIV is a virus. Viruses are not alive, by the way, because they're not a cell. They are just kind of like the sperm, like a capsule carrying a genome. And the genome in viruses can be made up of DNA or RNA. In the case of HIV, that genome is made up of RNA. And that RNA inside the HIV virus capsule encodes everything HIV needs to make more HIV and to propagate, but it doesn't have any of the equipment to do that. It doesn't have any factory to build those proteins. It just has the code. And so the HIV virus actually uses our cells' factories to make the HIV proteins and new HIV viral particles. When HIV infection happens, that RNA goes into our cell, becomes DNA, and permanently puts itself in our instruction book. It's essentially like walking into a library with, your own page of instructions, walking into a book and sticking it in and saying, now everyone who reads this book is going to read these instructions. And in those instructions, HIV says, the memo is always make more of me. And so what happens is then the DNA readers, our DNA readers read those instructions, make RNA that says make HIV's virus capsid, make HIV genomes, make HIV pieces, put them together in the cytoplasm, and then send them out into the world to do more things. And so we don't have any control. HIV doesn't care where in our DNA it goes. It just cares that it gets in our DNA and can do that. But it's always helpful if the HIV goes to a place that is actively being read. Remember, not every cell uses every part of the DNA. And so the cells that HIV infects are T cells, are using T cell specific instructions. HIV likes to hide in the T cell specific instructions to make sure that it gets read, but it doesn't always do it in exactly the same place. And so wherever it puts itself can create mutations or cause other genes to turn on or turn off that can change your disease outcome.
Thank you. Because we have to relate it home. We are are you able to stay on a few more minutes because there are many other very interesting questions? Jen?
Yes, I can stay a few more minutes. Yeah.
Okay. And when you're really tired of staying there, let us know. Annette.
Okay. Please elaborate on why there is much less genetic
By
testing
the
in
way,
Africa.
if Jen is not able to stay to answer all the questions, these questions, she's going to write down for us the answers so that we put them on the website, and many other people, including those who did not attend this webinar, can look at this as part of continuous learning. Yes.
Why is
Jen,
that?
you tell us when you're ready to leave.
Okay.
Why is there less genetic testing in Africa? Who benefits from this testing? How can we improve this situation?
So I think that's a better question for Africans. I would say, yeah,
Can any African on the call answer this question? Okay, let's go to the next one.
Okay. Clarify on the C trait in sickle cell. Help tell how it contributes to or otherwise.
I think that question has been answered, Annette.
Yeah, again, just briefly say, A is dominant, C is one recessive trait, S is another recessive trait. S causes sickle shaped cells, C causes cells that are still round, not sickle, but carry less oxygen. And that's the mechanism of action. The carriers do not have any of those impacts. Carriers look the same. The trait of a carrier is the same, or I shouldn't say trait because sickle cell trait is called trait, but if you are HbAs or HbAc as a carrier, your red blood cells still function like AA.
Is there gene therapy for HIV cure currently?
There are none that are authorized yet. There are more than 40 clinical trials that have happened in the world. About 15 of those are still ongoing right now for some kind of gene therapy to treat HIV infection. They have lots of different strategies for how to combat HIV. And there are more that are getting closer. So we're starting to see positive outcomes. The test in a gene therapy trial for HIV is, if you give the gene therapy, can the patient or the person living with HIV stop taking their antiretroviral drugs and not have the virus run amok? And the answer is we're starting to see the first patients in which that is happening.
So none of these studies is happening in low and middle income countries. That is why we need to have
Correct.
our own kind of gene therapy experts so that we can go from basic science, but we can also take products into translation from wherever they've been developed. So those who are still online, I think this is a very big gap which we all need to work together to close.
Okay. Does altering human DNA interfere with natural diversity of God's design?
That is a great question. So remember, we talked about changes that are inherited are called germline, and changes that are acquired are called somatic. Altering the human DNA right now is only done in somatic cells for human therapy so that it can't be passed from generation to generation. There was a case in China of a single researcher who had modified embryos using gene modifying tools. And that resulted in the birth of at least 3 babies whose DNA had been altered. And that was condemned worldwide as a step too far. I will say the discussion is ongoing. Right now, we have what is called a moratorium on changing the germline so that altering DNA could be inherited across generations. But there's a lot of different opinions about it. For example, if you were a set of parents and you learned, you had a child, let's say in your 20s, and then at the age of 35, one of you started showing symptoms of Huntington's disease, the brain degeneration disorder, and you knew that you already had a child and that child had a 50% chance of having an increase of having Huntington's. Um, you would want to decide whether or not to get your child tested and see what options there were to try and modify that outcome. But again, it's a dominant trait. So having the gene means the disease is most likely going to be expressed in that child. So if you have two parents that get screened when they get married, let's say, and they find out that one parent has Huntington's, would you want the ability to correct the Huntington's mutation in your child to get rid of that 50% risk? And the answer is that a lot of people with Huntington's disease would say absolutely. But the rest of us would say that's crossing the line. It would be a step too far to do that. And so we still have a lot to decide on. Again, there isn't one right answer here. Some people would also say, science tells us how we work, but faith tells us why we're here. And in science, people would say God gave us the knowledge to think about, to find out how our DNA works and how it's written and how it's organized. And a natural progression of that, in the true spirit of faith, is to do right by other individuals. And that means, doing what we can to help prevent them from having terrible, debilitating diseases that don't allow them to reach their full human potential. But there are also advocates on the other side that would say this is the natural order of things and it shouldn't be messed with. I think, again, we're a spectrum of very diverse human beings that is built in from our genetic instruction book all the way to our diverse biology that you see on the screen and we've heard about today. And so it's a personal decision, I think, that each individual has to make.
Thank you for that. The individuals with gene mutations that protect them from getting certain diseases such as CCR5 delta 32 for HIV. Would it be ethical to register such individuals as potential donors for gene therapy to treat others?
It's certainly something that people have looked into, but we also know something interesting. Remember, I said that some variants happen in certain geographies. The CCR5 delta 32 mutation that some people are lucky to be born with that allows them to resist HIV infection primarily occurs in European populations. But Europeans were not the first people to experience HIV infection. Africans were. We need to understand if there are Africans who also have CCR5 delta 32. And if not, we really need to think about why. It seems more obvious that a natural resistance mechanism should form in the first population infected with a disease or a virus such as HIV. If it didn't, it probably means that mutation, it's not that mutation never happened. It just means that mutation created other problems that resulted in those individuals not reaching reproductive age. And so if there is not delta 32 in African populations, we really need to pay attention to why not, because asking Africans to get delta 32 might create a bigger problem than just the HIV infection. So we have to look at all of it. Again, understand the gene, its functional outcome, and how it relates to our environment and also our behaviors to understand if it will be a meaningful therapeutic intervention or not.
Can mental health problems be related to genes?
Can you repeat that? Oh, mental health problems. I see. Yeah.
Problems be related to genes?
Absolutely. 100% yes. Both inherited and acquired genetics, but also behavioral problems, as well as other things that you can be exposed to in the environment. Yes, there are genes associated with neurologic conditions and mental health issues. I'll give maybe one example. I fall into the mental health category of attention deficit and hyperactivity disorder, ADHD. It means that the proteins in my neurons are not able to regulate slowdown, and they're super sensitive to dopamine triggers. And so I don't know the specific genes in my own genome that put me on that spectrum, but I do know that I have a family history of it, which means that it's most certainly related to genes that are passed down through my family. Learning how to live and work with that was a process that took some time. And so we know that, those inherited kinds of mutations can absolutely contribute to mental health and or spectrum disorders.
That's interesting. Should gene therapy be prioritized for rare diseases or also for enhancing general human traits? Please give your experience.
It should be used for any disease for which it can have a positive outcome. Rare, ultra rare, prevalent shouldn't matter. Of course, that is the moral and ethical answer. There is never, it is never morally okay not to treat a disease for which you have the capacity to treat it. That is my simple answer. Unfortunately, humans and societies are far more complex. They talk about money, they talk about, clinical access and other components that make it difficult to actually implement these things. I think in the gene therapy field, personally, because gene therapy is such a next generation sort of approach, the first diseases that we tried it in were really rare diseases that had no other options. Because the risk of this new crazy treatment causing a problem versus the potential benefits was in favor of the benefits. And that's because there was no other treatment for these diseases. Now that sort of proof of concept has happened, you quickly see the field moving away from those really rare incurable diseases towards things that will make the most money, or things that, could impact the people that vote for them, or the people that, care about that science or fund that science. But it's really important for us to remember how important rare and ultra rare diseases were to making gene therapy a success. And we cannot forget about those diseases. But again, I'll just reiterate, in my personal opinion, which is just my opinion, it is never morally okay to withhold treatment for an individual simply because of the disease they have being rare or ultra rare.
So Jane, there are many more questions, but we'll ask the last one, which is in the chat, and then we will compile everything and share with you so that you can other people can access these answers, because I also see some of the people already fell off. But for members who are still on, there is an evaluation form. Please fill it. Annette.
Yeah. The last one, smoking changes genes. How about us who have been on ART for years?
I think I don't know the answer to that question. No, none of the ART drugs are specifically intended to make changes to DNA. They're intended to stop HIV from making changes to the DNA. But, again, you take an ART drug, we often think like, oh, I take this drug and the drug just goes and does the thing that I needed to do inside my body. But sometimes that drug, it's got to go through your stomach. It has to be processed there, then get into your intestines, then be converted and transported out to your bloodstream. Sometimes it has to go to your liver first and be converted into a different form of the drug before it actually has the shape that it needs to do the thing you want it to do in the body. We call that metabolism. And there are so many genes, right? The genes of your stomach cells that make it function, the genes of your intestinal cells that actually process that drug, the genes of your liver that might metabolize it to its functional form, and then the cells the drug is actually working on, which are the T cells. And so there are variations in genes in every one of those cells and spaces that could potentially be impacted by taking those drugs. But the drugs themselves are not specifically designed to make changes to the DNA.
So thank you. We'll put our hands together again for a very educative webinar and answering most of the questions that we had and answering them very well. Thank you, Jane, and I'm sure everybody appreciates. We'll look at the evaluation forms. I think you have made DNA very simple, even to some people here who are communication experts. They agree with me that this has been very good. Thank you very much, Jane. And
Thank you so much, Sissy.
we will communicate by email. We would love your answers to be shared with even a wider audience. Thank you. Yeah, the next webinar, of course, is not for Jane, but it's on the 24th or 25th. We will send out the email as usual. Thank you.
Sissy, for the written questions, will someone send me a copy of the questions, or should I stay on and type answers now?
Oh, no. Annette will compile all the questions and send them.
Okay, thank you.
Thank you.